coming. Thanks, Ian and Nora, for inviting me. I'm very glad to be here. So, we are going to talk about species interactions under warming conditions, and in the last part of the talk I will show you some preliminary data on species intraspecific interactions under IPUC conditions. Basically, it is quite a new concept to include species interaction between climate change studies. And 10 biotic multipliers was introduced for the first time last year in this paper by Zartsniki et al. In Science, in which they basically say that species interactions have received very little attention in climate change studies, but in turn they play a key role in the functioning of ecosystems, even in some cases milding the effects of climate change. And in this paper they basically say that if climate change is widespread, we will see in the near future depression of top consumers within a community, a terrestrial community, and this will cascade down other trophic levels with the domination of herbivores and a very different primary producers community. Looking at these vertical interactions in terrestrial environments, some years ago we started looking at horizontal interactions within the food web community. So we were curious to see how competition could be a key factor in affecting climate change effects, and we started to look at this in small hybrids and fish. So basically my talk will be split into two main topics. One is how warming could affect interspecific competitions in grasses, and how an IPUC warm, according to ocean acidification, could potentially affect the intraspecific competition and sperm competition of another grassy species in the Mediterranean Sea. So we start in the first part with biological response to warming, which can take three broad forms, generally speaking. One is adaptation and plasticity that allow individuals to succeed in the same place. The other one is movement, both forward or vertical in depth or altitudes, and this is something that species can do to track tolerable conditions. And then the other response is a local extirpation or local extinction. At local scale, of course, species according to their characteristics can respond in different ways, and usually sessile mobile species are very influenced by these changes. We did see several mass mortalities according to deepening of thermoclining in several places around the world. For animals that can choose where to live, relocation becomes a valuable solution to track tolerable conditions. As a result, there is a great number of papers dealing with range contractions, range restrictions, and range expansions according to warming effects. And there is evidence in the literature also for fish assemblages, so the entire fish assemblages deepening, like in the North Sea and several other parts in the world. And another key effect is the way warming could affect the relative abundance of some species and the way this could affect also the magnitude of the intensity of the interspecific interactions. So we are, according to this, since climate should favor warm species over cold species at the same site, we are going to assist to a density increase at a local level, which in turn could be a problem of ecological antagonist if it's present of this species. So this suggests that warming could enhance and create novel interspecific interactions. This is what we basically see in some parts of the Mediterranean Sea, like native and invasive species interactions, and also intensify the strength of pre-existing interactions, like in the case we are going to see in few minutes. Of course, one meaning of coexistence for a species is to partition resource within the habitat, or to relocate to another habitat, so using different resources. Just a brief remarks on the warming in the Mediterranean Sea. We did notice a huge warming up to 1 degree Celsius, particularly in the eastern basin in the last 30 years. And we still in the western part of the basin, according to temporal series, we recorded a 0.66 increase on average of annual seawater temperature. As a result, isotherms are moving faster northward, and as a consequence of this, we are seeing the last 2 decades, I should say, an increasing amount of warm water species native from the Mediterranean Sea going northward, moving in the northern part, which is a concern also because we are limited, geographically limited in the north, so they have no space to move further. And so, for example, not for fish, but for mysids living in specific habitats, like marine caves, there are examples of local extinctions as a result of interactions between 2 mysid species, one of warm water adapted and the other cool adapted. And in addition to this, we are now recording a higher amount of alien species coming from the Suez Canal, which are now the most widespread and common and dominant species in shallow waters in the eastern part of the basin, like rabbit fishes. We did record now like 35 fish species of subtropical and tropical origin inside the Mediterranean Sea, and this year it was recorded the 1,000 non-native species, for the first time, 1,000 non-native species inside the basin. So the pressure from native species is quite a concern. And of course, we do have also subtropical species coming from the Gibraltar Straits. So in the literature, local literature, we basically speak about meridionalization process, which is the warm component moving northward, and a tropicalization process that is basically widespread in the eastern part of the basin. So most of the isotherms are moving at very fast rates, like it was seen last year, are moving northward, and we can see great evidence, even the public is quite concerned about this in the last few years. So very dramatic changes, particularly in the eastern side, but also where I live, which is here in Sicily, in the middle of the Mediterranean Sea. So we now have a well-documented we now have well-documented knowledge on the northward expansion, but we do not have any knowledge about the response of the cool water species inside the basin. So, and this could be a higher problem in the near future, since the predictions says that the Mediterranean will warm up from 2 to 3 degrees by the end of the century. So after this premise, we started working on using field surveys and control experiments on the potential interaction between two wrasse species, which are very common in coastal waters. And we first look at the entire potential range of both species in the Mediterranean Sea and in the Eastern Atlantic, just to look at patterns along the latitudes and longitudes. And then we started looking at patterns and vertical distribution in relation to different habitat characteristics, since one of our hypotheses was that they were being demersal species, and we'll show you them in the next slide. Space could be a limiting factor for them. And then we look at the segregation in depth and habitat at regional scale. Finally, I will briefly show you a controlled experiment we did manipulating temperature and density of the range extending species, the warm water species. So we choose to work with these two species, Coriciulis, the rainbow wrasse, and the ornate wrasse, Thalassoma pavo, on the right. They are small protogynous species. They share quite a similar diet and similar habitat preference. They do prefer rocky algal habitats. And in the last, they usually 30 years ago it was Thalassoma pavo, the warm water species, was only seen in the northern part of Africa and the southern part of Spain, Italy, and Greece. But in the last 20 years, it's moved northward, becoming established in the northern part, coolest part of the Mediterranean basin, which is the Ligurian Sea. With the Coriciulis show a more widespread distribution, uniform distribution within the basin, although in the last 5 years it was, there are some records of local extinction in the eastern basin. This is the relative likelihood of occurrence, and indeed they are present inside the Mediterranean Sea, but there are some differences according to these maps, because below this level, which is the Canary Islands, there are some sister species like Coris atlantica and Thalassoma newtoni, and there are no records for the British Islands since, I think, the 1890 or something like that. So we look at large-scale distributions, looking at the literature. We collected data from UBC, so underwater research census data on the literature, mostly on rocky algal habitat, encompassing the 95% of the potential range of Thalassonophile and almost the 60% of the potential range of the cool water species. And we did see that relative abundance is opposing. They have opposite trends, and this is highly correlated to temperature. So this is not surprising. Although the hypothesis that species are most abundant in the center of the range, it could not be applied to these process species, but this is not new in the literature. It's quite well known for some other fish species, I think also in Australia, Eastern Australia. So our first hypothesis was if a space is a limiting factor, since they are strictly benthic species, and they are territorial and gregarious, for coexisting, changes in the levels of bottom slope may affect the vertical distribution of this demersal process. I'm talking about bottom inclination because for the presence of the thermocline, both species are quite limited. At least the warmest water species is quite limited in the depth. They usually, when there is the seasonal thermocline, they do not go deeper than the seasonal thermocline. They do prefer to stay in warm water species. So maybe the coexistence with the potential ecological antagonist, the other cool water species, could be enhanced by habitat characteristics like topography. So we did surveys both in the Eastern Atlantic and Med Sea, so a lot of dives in those periods. And we collect data with standard transects at two depth level. Each one is below and the other one is above the seasonal thermocline. And we look at the distribution in four ecoregions, looking at if the different characteristics of the inclination of the bottom could affect the spatial distribution. So we basically see a consistent patterns in vertical segregation in high slope, in shallow water, high slope, which means with higher inclination coast. And this is consistent in different regions. But we did see also some differences according to a dominance even at deeper waters. This is maybe an intrinsic biogeographic difference because the Tipau, which is the green bar, and the warm species as its original species pool in this area in the Eastern Atlantic. So it is intrinsically higher in abundance. Keep going in looking at the patterns. We did the same, almost the same within the Mediterranean Sea, but we collected data along depth gradient from 1 to 36 meters, each 5 meters, in different, along the latitudinal gradient in the western part of the basin, from the Tunisian Plateau Libyan Sea to the Ligurian Sea, the coolest part of the Mediterranean Sea. We placed our transect in high slope conditions just to look at a potential vertical segregation of the two species. And we look at the difference. These are the results, and both species segregate at lower latitudes. So as soon as the density of the world species gets higher, then it seems that the cool water species is pushed down in deep waters. And of course, in the Ligurian Sea, where the warm water species is present only in the last 20 years, we did record only few amount of species only in shallow waters, few amount of individuals only in shallow waters. Then we did the literature just to assess the generality of these patterns, and we exploited own data sets, and we collected data from almost 400 sites along the Mediterranean coast at 2 depth ranges, using the same, focusing on the same technique and different years from 1992 to 2010. And these are results in shallow waters. And it is evident how the warm water species in orange is the widespread and dominates in shallow water in the southern part of the basin. This was not like this some years ago. And while the cool water species is still dominating in the northern part. We have differences, although the cool water species is less present in deeper waters. So we started thinking about this at these patterns, and so we wanted to understand why, if the cool water species is pushed down, maybe, of course, they will need another thermal environment, but maybe it's a less favorable habitat, but we have no tools and facilities to further assess this in an experimental way. And we start thinking about looking at, instead of a vertical shift, to a potential horizontal shift of the 2 species within the same thermal environment. So if the warm water species is displacing the cool water fish to a less preferred habitat, this was our question within the same environment. And we look at the abundance on a latitudinal gradient of the 2 species in the 2 most abundant shallow water habitats in the Mediterranean Sea, the seagrass Posidonia oceanica, and then brown algae on growing or rocking many Cystoceria species. We did use the same technique, and same results as seen before. So segregation and different domination in shallow waters. We focused on shallow waters between north and south. So the warm water species is more abundant in the south. The cool water species, of course, is more abundant in the north. But as soon as the warm water species relative dominance is higher in the south, we did notice a higher abundance of the cool water species within the same thermal environment in a less preferred habitat, which is the seagrass habitat. So we tested this in large arenas, giving basically to the cool water species a pairwise choice between the 2 habitats, the preferred habitat, the algal habitat, and the seagrass habitat. And we assessed its relocation, manipulating temperature and the density of the warm water species. So trying to mimicking what could happen in a warming scenario. We did collect a lot of individuals to do these experiments. Basically, the arenas were covered with two different habitats, the grass and the macroalgae, and we put one time, two times, three times relative dominance of the Bormote species inside the arena to assess the relocation response according to two different temperature ranges. Present day scenario, which is supposed to be ideal for both species, and a projected scenario, just to look at density dependence response of the cool water species to increasing abundance of the warm water species. This is the experimental design I want to show because it's quite difficult maybe to understand in the results. So we add two levels of temperature, present day and projected, three levels of density of the cold warm water fish, and 6 different habitats. Since it is a pairwise choice, we look independently. As an example, AES means that in the pairwise algal habitat, seagrass habitat, we look at the algal, the density within the algal habitat, and SA is the opposite. So in the pairwise choice between seagrass and the algal habitat, we look at seagrass. And the response variables we compared was the number of individuals present on each habitat independently, and the habitat permanence, and some behavioral activities measured as percentage time. Okay, quickly the results. We did not notice any difference, although some trends are evident in this case at projected warming, at least for the first two density treatments. And we did notice a shift, a relocation to a less preferred habitat at projected conditions at the highest level of density of the warm water species. So the cool water species relocated to another habitat when the density was particularly high. So was not related to temperature in present day conditions, this response at the same density treatment. But if warming and density relative dominance of the warm water species act in concert, the response was quite variant, and the similar response was observed on habitat permanence. We also did some behavioral analysis, but results are quite variable. Maybe the only interesting thing is that resting activity is higher at warming conditions, projected warming conditions. We wanted to look at the physiological responses of the cool water fish, but Paolo Domenici, which was a previous PhD student of this university, will start the experiment next week. So in the meantime, we will publish these results. Another topic, and there are just preliminary observations we did in the last 2 years, taking into account the potential effects of IPCO2 world on sperm competition in another Mediterranean race, Symphodus ocellatus, the ocellated race. I'm not going to talk too much about ocean acidification. We have basically an increase of the concentration of CO2 in the atmosphere. This results also in a higher PCO2 in the ocean and a lower
pH. And we used the carbon dioxide bands to look at the potential differences and how high PCO2 would affect the sperm competition, intraspecific competition in this process. Maybe most of you were present in the talk Monday gave on Monday, so maybe you know that there is quite a great number of studies, mostly made by him on the effects of CO2 on the behavior and olfactory responses of fishes, specifically mostly at larval stage. So we used these gradients in the island of Vulcano, which is in Sicily. We did measure some basic carbonate chemistry, and we focused on the intraspecific competitions of this Russell, which has a very nice behavior, particularly males. They build dish-shaped nests with algae, and they guard the eggs. They occur mostly in shallow waters, and this behavior is by males is basically we have a nesting male which spends most of the time during the spawning season to build an algal nest, and then invites females to spawn in his nest. We have a bunch of other males, like a satellite, which is in charge of inviting females in the nest, and also chasing sneakers males, which take profit of simultaneous parasitic spawning and take profits of the female presence in the nest to fertilize the eggs. This kind of spawning behavior is not uncommon in fish. According to data from I think 5 or 6 years ago, this kind of simultaneous parasitic spawning behavior is well documented for 140 species. And the dominant family is the Labrida, the Rosses. So let's look at the results. We basically look at the nest during the spawning season. We marked these males just to be sure that the male was the same during the spawning season, because there is can occur a nest takeover according to aggressive behavior between different males. And we filmed them during the spawning season in high PCO2 and low PCO2 ambient conditions. These are the main actors during the spawning season. So we first look at the maximum number of females within the nest and the total number of females. Maximum number of females was counted on 15 seconds within 10 minutes recording, and total number of females within the 10 minutes recording. We did this for a couple of years, and the same was done for sneakers, because satellites are not so common in this. We found only 5 nests out of 100 with satellite males. Sorry. Before showing you the preliminary results, I'm going to show you what happens in these two conditions. We are not sure about the mechanism yet. So we have basically the nesting male, the female. He invites her to come over the nest, and this is a sneaker that takes profit of this condition, but in this case was late. Look at the fish. Wow.
Wow.
So sure you will get crazy about this. This is not uncommon. This is a situation in ambient seawater, which is only 300 meters apart from the site we saw before. And I'm showing you two extremes. So of course things are. And so these are the results we are looking. We have looked at in the last 2 years. So basically, fertilization events by the sneaker male are the dominant conditions in a high PCO2 world, while the opposite occurs in ambient seawater. This is in accordance with the previous literature from the Corsican population. So this difference is quite common for these species. This is not. We are not sure about the mechanism yet, but what we are doing right in these days is we have collected eggs at the late developmental stage, and we are now doing paternity analysis on these eggs just to assess if these higher fertilization events, extra pair fertilization events, are confirmed by the offsprings. And this is basically the end. Thank you.
Did you look at fertilisation success? Were you able to look at that in the eggs that you collected? Was there any difference in the proportion of eggs that were fertilised?
No. Yes, we looked, but there was no difference. But we don't know. Yeah, the fertilization looked similar. I need to, but because we have almost 6,000–7,000 eggs in each nest, so a student of mine is right now analyzing the data, but by looking it seems the same.
Yes, I would expect that.
So for your experiment on the temperature effects on the competitive between the water and warm water species, what I didn't catch that. What are the mechanisms that competitive displacement of the cold water species from the habitat? Are they more become less competitive in getting food, or?
We did not test competition for food, but we test only competition for space. I gave no details on some preliminary experiments I did to set up the final experiment, but giving them a choice between two different habitats, then they do prefer one of the two in the arena. So basically increasing the density of the warm water species, we did notice a different behavior of the cool water species under warming conditions, a higher resting rate, and we noticed that the only way to be safe from aggressions from the warm water species was to rest underneath the seagrass. And so basically, by the moment we look at this only from a behavioral point of view, looking at space and segregation in a limited space, which is in a small arena of 50 square meters.
I was just curious if you could comment on what you think the implications are for the last results you showed. So if in more acidic waters you get more sneakers, what are the implications for the species for that? So are sneakers typically a lot smaller, and is that a bad thing for the species?
Well, we don't know, but for sure evolutionary implications and higher sneaker and extra pair fertilization is indeed, and that, there are some examples for these species, and indeed a concern in an evolutionary perspective, and well, that's basically what I can say.
Oh, I was going to ask the exact same question. I guess I can follow it up though. It seems like there's going to be a balance in terms of natural selection between the sneaker strategy and the nesting male strategy. Is that balance going to shift if it's becoming easier to become a sneaker, or are you just going to end up with all sneakers?
Well, I'm sure I got your question. A balance, you mean in ambient seawater?
Yeah, so in current conditions, there has to be some selective advantage to being a sneaker at a certain density,
Yeah.
and some advantage to being a nester. And if the relative advantages are changing, and it looks like they are, does that mean that the proportion of sneakers in the population is going to increase or decrease?
I don't know, because maybe I can show you this graph. This one. Basically, the sneakers are 1 year old, and as soon as they get the second year, they switch to satellite and or nesting male. Most of them will be satellite according to authorities' data. So I don't know if this will increase the amount of sneaker male, because I don't know if the sneaker behavior is genetically